So, our topic is discovery of DNA. 1869, Friedrich Miescher discovered nuclein. He found a substance inside a nucleus and basically isolated it and termed it nuclein. And in 1878, Albrecht Kossel isolated non-protein part of nuclein, and what he discovered was there was a nucleic acid, and 5 bases: adenine, thymine, guanine, cytosine, and uracil. So it's pretty amazing that in 1927, a Russian scientist, Nikolai Konstantinovich Koltsov, said something which is very interesting. Inherited traits would be inherited via a giant hereditary molecule, which would be made up of 2 mirror strands that would replicate in a semi-conservative fashion using each strand as a template. Don't forget that this is before we know the structure of DNA, and he is basically hinting that आपका hereditary molecule क्या होगा. Till that time, they only knew that there are 4 bases and there is nucleic acid, but we have no clue whether it's a hereditary molecule or not. So it was, let's say, a very interesting fortune telling. And 1928, Frederick Griffith basically established that there is some sort of transformation which happens in bacteria. So basically, at that time, pneumonia was very common, and there were 2 strains of pneumonia. One was a rough strain and other was a smooth strain. So rough strain was non-virulent, while smooth strain is virulent, and because of it has basically a glucose shell on it, or basically let's say carbohydrate shell on it, basically which makes it virulent because it escapes your immune system. So in first image, what you see is that when you inject mice with a rough non-virulent strain, it doesn't die, it lives. But when you inject a smooth strain, mouse dies. Basically आपने establish कर दिया कि इसके अंदर से जो strain disease cause करता है वो कौन सा है. But when you heat kill a smooth strain, mouse lives. So basically एक और चीज साबित कर दी कि अगर आप pneumococcus को अगर आप heat kill करें तो it cannot cause disease. But now this is very interesting. But if you mix a rough strain and heat kill a smooth strain, mouse dies. यानी कि जब आप रफ स्ट्रेन डाल रहे हैं अकेला तब भी माउस नहीं मर रहा, जब आप हीट किल्ड स्ट्रेन डाल रहे हैं तब भी माउस नहीं मर रहा. तो क्या वजह है कि जब आप जिन दोनों स्ट्रेन से माउस नहीं मर रहा उनको मिक्स करके डाल रहे हैं तो वो मरने लग जाता है. सो अल्टीमेटली हाइपोथेसिस वाज कि कोई चीज ट्रांसफर हो गई है हीट किल्ड स्मूथ स्ट्रेन से रफ स्ट्रेन को, जो अल्टीमेटली जिसने रफ स्ट्रेन को भी विरुलेंट कर दिया है. और वो चीज नाउ वी नो क्या थी, डीएनए, जो अल्टीमेटली ट्रांसफॉर्म हो जाता है रफ स्ट्रेन को. So in 1944, Avery, McLeod and McCarthy basically isolated DNA. On your screens, you can see a white glass, and basically they developed a protocol using phenol to precipitate DNA. So in its bottom, you can see a white precipitate, and that is DNA. Now, but the final conclusive evidence came from an experiment done by Hershey and Chase. On your screens, you can see a bacteriophage. So bacteriophage has an outer protein shell and the core is made up of DNA. And bacteriophage infects bacteria and survives inside it. So इसको reproduction के लिए bacteria चाहिए. So the evidence would be कि अगर ये वायरस कौन सी चीज़ बाहर छोड़ता है और कौन सी चीज़ वायरस बैक्टीरिया के अंदर जाती है so that it can reproduce. Let's see what they did. So what they did was they radio labeled outer coat of protein, sulfur in it basically because cysteine contains sulfur, and they also radio labeled DNA which was inside. So what you see in these images, which is very clear, what they found out was that all sulfur containing shell was stayed outside a bacterium while DNA went inside. In this way, they were able to conclusively prove that basically the material which goes inside a bacterium is DNA. So, what you are watching on the screen is known as photo 51. Basically, it's an X-ray crystallograph of type B DNA. And one of the students of Rosalind Franklin basically took that picture, but nobody knew how to explain it. And there was a debate between Maurice Wilkins, who was student of Rosalind Franklin, whether it's a helical structure or some other type of structure. So Maurice Wilkins was of the idea that it's a helical structure. He showed this image to Watson and Crick from University of Cambridge, and they proposed that it's a twisted ladder-like structure where sugar phosphate backbone is on the outside and different bases are paired with each other. So they published their findings in the most reputed journal at the time, and which still is, Nature. And there is a funny incident with it. This is the only non-peer-reviewed article. When the editor of the journal saw their findings, he was so sure that if he would send it for peer review, the information would be leaked. So what he did was that he just basically published without peer reviewing it. And that's how basically DNA bases are paired with each other. So adenine and thymine have 2 hydrogen bonds between them, and guanine and cytosine have 3 hydrogen bonds between them. So यहाँ से basically once DNA structure was solved, एक नई era का आगाज होता है कि can we edit the DNA, and that's we'll see. Thank you.